Hello, today we are going to talk about the adaptive immune system. Our adaptive immune system targets specific pathogens. We're going to talk about the major cells that play a role and they are actually part of our adaptive immune system, such as our B cells and T cells. And we're going to talk about how do they target and how do they bind to these pathogens. Your adaptive immune system gets deployed when you have, for example, you've contracted an infection or if you get vaccinated. In either cases, your adaptive immune system is going to go ahead and help you build immunity against that pathogen. The first time you contract an infection, we refer to that as the primary response. And the primary response is going to be much slower compared to your secondary response. Your secondary response is when you go ahead and you contract the same pathogen for the second time. That could be months later or it could be years later. Your secondary response is always going to be faster compared to your primary response. And the reason why it's always going to be faster the second time you get exposed to the same pathogen is because you have developed memory cells, and you develop those memory cells from the very first time you've contracted that infection. And those memory cells actually they do live for a long time, and so therefore having these memory cells will definitely help you attack that pathogen faster the second time around. We divide our adaptive immune system into two major parts, our humoral immunity and our cellular immunity. The term humoral means blood. The cells that would produce antibodies, those are our B cells. And when B cells produce and release those antibodies, the antibodies are going to circulate within your blood's plasma. And so because they are going to be circulating freely in your blood's plasma, they are part of your humoral immunity. And again, humoral means blood. These antibodies, they are going to help combat and launch an attack against pathogens. And you can refer to these pathogens or these foreign molecules, we can use the term antigens. And we're going to talk about how antibodies go ahead and combat these foreign molecules that could be within your body. B cells, by the way, they are a group of white blood cells. They are under the group of white blood cells known as lymphocytes. We actually have two types of lymphocytes. We've got our B cells and our T cells, but our B cells are the ones that produce antibodies. Those B cells, they are made in your red bone marrow. And that's why they actually refer to it as B cell, since it's made in the bone marrow, and actually it's also going to mature there. So when the B cells are first made, we consider them immature. They actually have to go through a process where they become mature and they have to recognize the difference in terms of what to attack and what not to attack, because you don't want to develop an autoimmune disorder where your own immune cells, your own antibodies are attacking your cells. So they have to go through a process where they have to know again what they're supposed to attach to, and the only thing they're supposed to attach to really it's those foreign molecules known as the antigens. So they have to be able to recognize these antigens and they have to be able to make those antibodies. The other part of our adaptive immune system, we refer to it as our cellular immunity. And the cells that play a major role in our cellular immunity are our T lymphocytes. So as we said earlier, our lymphocytes, we've got two major types of lymphocytes. We've got our B cells that produce the antibodies, and then we've got our T cells. We actually have several types of T cells, and we're going to discuss two of them in this lecture. We're going to talk about our helper T cells, and we're going to talk about our cytotoxic T cells. We're going to talk about how do they go ahead and launch an attack against these antigens that might be within your body. Now, where do the T lymphocytes, where are they made? They are made within your red bone marrow. But for them to be mature, so they also have to go through a process where they have to be able to recognize what to attack versus what not to attack, because as I said, you don't want to develop an autoimmune disorder. So they head on to your thymus gland, which is a gland on top of your heart, and this is where they become mature. So your T lymphocytes, they're made in the red bone marrow, and then they head on to the thymus gland, and this is where they become mature. And that's why they refer to it as the T lymphocyte in reference to the thymus gland of where they become mature. On the surface of the T cells, there are receptors, and those receptors, they are actually going to be pretty important because those receptors will have to bind to these pathogens or antigens, and this is how the T lymphocytes will actually get activated. And we're going to talk about it in more detail. Once T cells get activated, some of them are going to start producing chemicals known as cytokines. They're not going to produce antibodies, so T cells do not produce antibodies. They could produce cytokines, and we're going to talk about the importance of cytokines in just a few. So this image right here shows you that to make those B cells and T cells, it all starts with some stem cells that are found in the red bone marrow. And actually another location of having those stem cells that can differentiate into B cells and T cells is also in the fetal liver. Once those stem cells, they go ahead and they start differentiating, they will differentiate eventually into the B cells and the T cells. And as I said, those stem cells are either in your red bone marrow or in the fetal liver. After the B cells and T cells are made, they will have to go through a process where they need to become mature. The B cells, they're going to be made in the red bone marrow, and they stay in the red bone marrow to become mature B cells. The T cells, on the other hand, they are going to be made in the red bone marrow, but they head on to the thymus gland, which is a gland on top of the heart, so that they can become mature. Once you have a mature B cell and T cell, those mature cells, they're going to migrate to your blood, they're going to migrate to your lymph nodes, your spleen, and after they migrate to these regions, that's when they're going to go ahead and start getting to work by attaching to foreign antigens that might be within your body, and they're going to start protecting you. So we will talk about how are they going to, again, bind to these pathogens. Our cellular immunity, again, the major player of our cellular immunity are our T cells. Those T cells, they can definitely attack multiple different foreign antigens found inside of your cells. So if a cell is infected by a virus, or if there are some fungi and parasites, again, those T cells can definitely help attack them. The humoral immunity, we talked about how your B cells are the major player of the humoral immunity because they're going to be the ones producing those antibodies, and these antibodies are going to also help attack those foreign antigens, and antibodies are usually they usually help you out by not entering your cells. They're going to do all the work outside of the cells. They're not going to affect the cells, and they can actually attack bacteria and toxins. And anyways, bacteria is known to stay outside of your cell. It doesn't penetrate deep into your cell versus a virus. A virus, we know that when a virus attacks, it likes to inject its nucleic acid deep into your cell. But with antibodies, they can go ahead, get to work, and they can start attacking these bacteria and toxins, even though they are not deep within your cells. Now, if we take a look at a bacteria cell, the bacteria cell, it usually has antigens on its surface. And so this whole thing is an antigen. The part that you're going to see sticking out of the bacteria surface, we refer to it as an epitope. So an epitope is really a part of the antigen that's going to be found on the surface of the bacteria. Now, as we said, our B cells produce antibodies. So here are a few antibodies that you can see right here. And antibodies, they are pretty specific. So why do we say they are very specific? Because that part of the antibody right there, known as the variable portion, needs to fit with, and needs to kind of like interlock with those epitopes, with these antigens on the surface of the bacteria. If they are not the same shape and they cannot interlock, then those antibodies are not going to be able to bind to these bacteria right here. So you can see that antibody A, for example, is able to bind to that triangular shaped antigen. It's not going to be able to bind to these right here, to these epitopes, because it's not the correct shape. So we produce many different antibodies, and again, all of these antibodies, they are going to be pretty specific. Now, the antibodies, they look something like this, something like a letter Y. Antibodies are proteins, by the way, and the protein looks something like this. It has some heavy chains and light chains that are bound by some sulfur bonds. We refer to it as disulfide links. Part of the antibody is constant, so it doesn't change. So basically this like stem region right here, it's always going to be the same for all the antibodies that you have in your body. And then there's a portion of the antibody that is up here known as the variable region, and this is the part that's going to change from one antibody to another. And this is the portion that would bind to the specific antigen that you might have within your body. There are actually 5 classes of antibodies that we are going to talk about. Another name for antibody, by the way, is immunoglobulin. So that's why immunoglobulin, we go ahead and we abbreviate it as IG. So immunoglobulin, antibodies, they're the same thing. And so we have 5 groups of those antibodies, or these immunoglobulins, that we are going to talk about. The first one is immunoglobulin G, antibody G. This actually makes up 80% of the total antibodies in your blood's serum. You can find those antibodies in your blood, in your lymph nodes. You can find them, in your like lymph fluid, in your intestines. These antibodies, they have the ability to cross the placenta. So the mom, when she's carrying the baby in her placenta, the mom can actually pass some of her own antibodies on to the baby to protect the fetus, because the immune system of the fetus wouldn't have developed yet. Antibody G can also help trigger and activate the complement proteins. We kind of talked about the complement proteins previously in the other video. So those antibodies, they can help enhance and trigger those complement proteins. They can enhance the phagocytosis process. They can neutralize toxins and viruses. And actually, we're going to talk about how do they neutralize the virus. They basically start surrounding the virus, and we will talk about it. There's another antibody known as IgM. This makes about 6% of your antibodies in your serum. It's larger than the antibody G. And so therefore it's not going to be able to cross your tissues. The mom cannot pass it on to her fetus, so it's not going to be crossing the placenta, and that's because it is much larger in size. Now your IgMs, they tend to cause clumping of your cells, clumping of the viruses. They are also involved, by the way, in they respond to your AB blood transfusion. So if someone is getting blood transfusion and they get the incorrect blood type that's not compatible with theirs, the IgM is going to start clumping up the red blood cells. They can also activate those complement proteins that we've previously discussed. Antibody M or immunoglobulin M, they are one of the first ones that would actually respond to an infection. So if your immunoglobulin M's are pretty high in number, that could indicate a recent infection or an active infection. Immunoglobulin M's, they don't last long in your plasma, so they are pretty short-lived. They would be destroyed, and then again, you would have to make more if you want to attack again viruses or clump cells, for example, like red blood cells that are not compatible with yours. You would have to make some more. Immunoglobulin A's, they make about 13% of your antibodies. You can find them in mucous membranes and saliva, tears, breast milk. That's why actually when a baby is born, it's pretty important that the baby gets that first batch of breast milk from the mom, because it will contain lots of antibodies that can help the fetus, that can help the baby, because again, their immune system wouldn't have developed yet. Immunoglobulin A helps prevent the attachment of the microbes to mucous membranes. There is another antibody known as immunoglobulin D, and this one makes about 0.02% of the total antibodies in your plasma. You can find it in your blood, in your lymph fluid, even on the surface of your B cells. So on the surface of your B cells, you do have immunoglobulin D. In terms of their function, we just know that it's going to help out in your immune response. Especially, it's going to help assist that immune response on your B cells. But otherwise, we don't know too much about it yet. We also have immunoglobulin E. That's another antibody. It makes about 0.002% of the antibodies found in your plasma or in your serum. They're actually found bound to your mast cells, the surfaces of your basophils, or they could be circulating in your blood. They do help us respond to allergic reactions. Their concentration increases when you're having an allergic reaction or when you are having a parasitic infection. Their numbers could also increase because those antibodies, they do help break open the parasitic worms or basically a parasitic infection if you have one. Um, Let's talk about our B cells. Now, our B cells, as I said, they're made in our bone marrow. They mature within our bone marrow, and then after they mature in your bone marrow, they head out so that they can start helping out. Now, the B cells, even though they are mature when they leave the red bone marrow, they're still considered inactive. And so if they remain inactive, then they're not going to help us out. So we need to activate them. Now, to activate a B cell, that can actually be accomplished in several ways. One way to activate a B cell is for that B cell to directly bind to an antigen, directly bind to a foreign pathogen in your body. Now, we produce many different types of B cells. What's different about the B cells that we produce are those antibodies that are found on the surface of the B cells. Let's say that here is the stem cell, and the stem cell has made the B cells. As I said, those B cells, they will be made in your red bone marrow. They will mature in the red bone marrow. After they mature, they head out. So they head out to your blood's plasma. They head out to the lymph within your body, that lymphatic fluid. But they are still considered inactive, as I said. For them to get activated, one way to activate them is they need to bind to some foreign antigens in your body. Now, notice how these antigens, for example, they were only able to bind to this B cell right here. This B cell right there, they were not able to bind to it. That's because the antibodies have to have the correct, again, like shape to bind to these antibodies. So you have to have a perfect fit, basically, between the antigens and the antibodies on the surface of the B cells. That's going to actually activate the B cell. So when a B cell binds directly to an antigen, that actually triggers the B cell to get activated. Once the B cell is activated, it can actually proliferate. Proliferate meaning it's going to make more of itself. So it starts to increase in number. And then after it increases in number, some of the B cells are actually going to be considered memory cells. And those memory cells, they can live for a pretty long time. They will never forget how a certain antigen looked like. And so these memory cells can actually eventually help launch a faster attack against these antigens. Another type of cell that will result after a B cell proliferates, it will start to differentiate into memory cells, and then the other cell that it will differentiate into are cells known as plasma cells. These plasma cells, their job is to start releasing antibodies. So it can start releasing the antibodies. And by the way, the antibodies that are released by the plasma are the same antibody on the surface of the B cell. And so the reason why these plasma cells are going to be producing a bunch of antibodies, because we know that these are the antibodies that were able to bind to the antigen in the first place. And so these antibodies will actually be released so that they can start circulating your blood's plasma. So if we take a look at this image right here, we can see a B cell, and you can see those receptors on the B cells. And again, these are actually antibodies. A B cell can actually carry more than 100,000 antibodies on its surface. And these antibodies, they will need to bind to the antigens. It will have to be a perfect fit between the antigen and the antibody on the surface of the B cell. Now, B cells are known to take in those antigens, so it's able to suck it inside of it, and it can actually start to break it down. And after it breaks down that antigen that it just took in, it will go ahead, kind of like spit it out of itself, and it's going to bind it to a receptor, another receptor found on the surface of the B cell. And it goes ahead and it presents it on that receptor right here on its surface. Once it goes ahead and releases a piece of that antigen, and it goes ahead and it sticks it on its surface, specifically within that MHC receptor, a T cell known as a helper T cell can actually bind to this like whole complex right here that you are seeing, and that helper T cell starts to release chemicals known as cytokines, which can help activate the B cell. So as I said, there are multiple ways of activating a B cell. One way is for the B cell to directly bind to an antigen. Another way of activating a B cell is via helper T cells. You can see right here, the helper T cells will bind to the B cell, and once they go ahead and they have that contact, the helper T cells are going to release chemicals known as cytokines, and these cytokines, they're going to help activate the B cell, and then eventually the B cell, after it's been activated, it will differentiate into memory cells and into plasma cells. And as I said, the job of the plasma cell is to go ahead and release a bunch of antibodies. Now, once the antibodies are released by the plasma cells of your B cells, now how do these antibodies help us out? Antibodies are not going to launch any direct attack, by the way. So they're going to help you out in an indirect way of how to get rid of these pathogens. One way is a process known as agglutination. Agglutination means clumping. So if there are bacteria, let's say inside of you, those antibodies that are going to be released by the plasma cells, those antibodies, they stick to the surface of the bacteria to the point that these bacteria cells, they start to clump together. Now, that process of clumping up the bacteria is not going to kill the bacteria. So how is the bacteria going to die eventually? That clumping right here, that agglutination that you are seeing, that's going to attract phagocytes. And when phagocytes are attracted to that site right here, the phagocyte is going to go ahead and swallow these bacteria right there. Now, another way for antibodies to go ahead and attack, other than clumping and causing agglutination, another thing is these antibodies, they can start binding to the surface of the bacteria, but again, that's not going to kill it. When those antibodies bind to the surface of the bacteria, that's going to help activate a certain protein known as the complement protein. And we talked about what complement proteins can do once they are activated in the other video. Another thing about antibodies is they can start coating the bacteria all over, or they can start coating viruses all over. And again, that's going to grab the attention of the phagocytes to come and engulf this bacteria or this foreign antigen. Another way is a process known as neutralization, where the antibodies, one more time, goes ahead and circulates the whole entire pathogen. Now, when the antibodies circulate, or basically, I should use the word coat, when they go ahead and they coat the whole entire pathogen, how's that going to be helpful? When they coat it like this, they are actually going to, again, attract the attention of the phagocytes to come and check out what's happening right there. And so, as you can see, antibodies do not directly kill. They don't directly kill the pathogen. What they do is they either clump up the pathogens or they coat the pathogens. And in either cases, when they do that, they're going to grab the attention of the phagocytes that are going to kill them. Again, kill these pathogens. Or these antibodies, they stick to the bacteria and then they help activate and turn on these complement proteins, which will launch the direct attack against the bacteria. So this is how antibodies work. And again, those are the antibodies that are going to be released by the B cells. Now, as I said, if you first contract an infection for the first time, it's usually going to take a while before you have a good amount of antibodies to launch a serious attack against those pathogens. It could take up to 10 to 17 days before you have a good amount of antibodies to get rid of that infection if you have it. Now, if it's your secondary immune response, so if it's your second time or even third time, etc., of contracting the same infection, it's definitely going to be faster the second time around. And we kind of mentioned it briefly. The reason why it's going to be faster the second time around, it's because this time you would have memory cells. So because you will have those memory cells, instead of waiting about 17 days before you get a good amount of antibodies to launch like a strong attack, you will have a good amount and a high amount of antibodies just within 2 days. So there's a huge difference between having a good amount, a large amount of antibodies in terms of 2 days versus if it was your primary response, you would have to wait around 10 to 17 days before you have a good amount of antibodies. So that's your, that's what's going to happen if we're talking about your secondary immune response. So as I mentioned, a B lymphocyte can either be activated directly by binding to a foreign antigen, or it can be activated after it binds to a helper T cell. Once that B cell binds to the helper T cell, the helper T cell releases a bunch of cytokines, and these cytokines will help activate the B cell. Once the B cell is activated, as I said, it's going to start making more of itself, and then it's going to start to differentiate into either memory cells or into plasma cells. And these plasma cells, their main job is to start releasing those antibodies that can help you launch a good attack against antigens. As I said, if it's your primary response, this is going to take a while. So it usually takes, there's a lag time, 3 to 6 days before you go through the differentiation process, before you start making, some plasma cells, some memory cells, and then it will take another, several days. For you to have a good amount of antibodies to launch a strong attack. So it usually takes around a total of like 17 days or so to have a good amount of antibodies. Now, let's say you get infected by the same pathogen. Who's going to help you out the second time around? Your memory cells. So your memory cells are going to be able to recognize that pathogen, and those memory cells will eventually differentiate into plasma cells and into some more memory cells. But the second time around, you will actually be able to produce so many antibodies in just about 2 days. So instead of 17 days where you have a good amount of antibodies, this time, the second time around, you can actually produce a high amount of antibodies just in 2 days. So that's why, again, we say that your secondary immune response is going to be much, much faster compared to your primary immune response. Now, on the surface of your cells, there are receptors known as MHC receptors, which stands for Major Histocompatibility Complex. Now, we actually have two types or two groups of these receptors. We have what's known as class 1 MHC, and we have class 2 MHC. Class 1, those are receptors that are found on all of your cells that have a nucleus. So any cell in your body that has a nucleus will have this receptor on its surface. Class 2, you only find it on top of your cells that are known as antigen presenting cells. What are example of antigen presenting cells? Well, those are your B cells, your dendritic cells, and your macrophages. So these three cells right there are going to have class 2 MHC on their surface. Now, as I said, we have a set of receptors known as the major histocompatibility complex receptors. And we have two groups of these receptors, MHC class 1 and MHC class 2. MHC class 2 receptors, they are found on the surface of cells, and these cells are B cells, dendritic cells, and macrophages. Those three cells are known as APCs. APC stands for antigen presenting cells. Now, why do we call those three cells right there antigen presenting cells? Because those cells are actually going to present a piece of the foreign antigen within their receptor. Why a piece of foreign antigen? Because those three cells right there, they are known to engulf a pathogen. And after they engulf it, they are capable of breaking it down, taking parts of the fragments of the pathogen that they've just broken down, and they go ahead and they release it out and they stick it on their surface, specifically within their MHC2. Now, who are they going to present it to? They are going to present it to a group of T cells, and we will discuss that shortly. So let's say that here is a bacteria. You have a macrophage. So let's say that this cell right here, it's a macrophage. Now, macrophages are known to be phagocytic cells, meaning that if a macrophage encounters a bacteria, what it's going to do is it's going to go ahead and engulf that bacteria. After it engulfs that bacteria, it's going to start chopping it up inside of itself. So let's say that this bacteria, this macrophage was able to go ahead and engulf it. Once it does, it's going to start breaking down the bacteria into little pieces because it's going to be able to digest it inside of it. And then it takes a piece of that bacteria, takes a piece of that antigen, and what is it going to do? It's going to go ahead and it's going to stick it on its surface. Specifically, it's going to stick it on its MHC number 2. And that's why we call it an antigen presenting cell, because what it's doing right now is it's presenting a piece of that antigen on its surface. Now, who is it going to be presenting it to? It's going to be presenting it to T cells. Now, we discussed how B cells have the ability to bind to an antigen directly. A T cell, on the other hand, cannot bind directly to an antigen. A T cell needs to bind to a cell that's going to be presenting the antigen to it. Now, which T cell is that MHC2 going to be presenting that piece of antigen to it? A type of T cell that we have known as your helper T cells, or we refer to them as your CD4 T cell. So let's go ahead and discuss that in more detail. So let's say that this cell right here is representing a macrophage. Macrophage are phagocytic cells, meaning that if they encounter a foreign antigen, such as a bacteria, they're going to be able to engulf the bacteria. Once they engulf the bacteria, they're going to start breaking it down. So let's say right here, this is representing the broken pieces of the bacteria, pieces of the bacteria's antigen. On the surface of the macrophage, there is a receptor, and that receptor is known as MHC2. What the macrophage does is it takes a piece of that antigen of the bacteria, and it goes ahead and it presents it on its surface, and it fits it into its MHC2 receptor. Now, who is it going to be presenting it to? The macrophage is going to be presenting that piece of bacteria antigen to one of your T cells. Which T cell? That would be the Th cell, or we refer to it as the helper T cell. Another name for the helper T cell is CD4 T cell. Now, why do we call helper T cells CD4 T cells? Because on their surface, they do have a protein receptor known as the CD4. On their surface, they also have another receptor called the T cell receptor. Now, what happens is this whole complex that you are seeing right there, it's actually going to interact, and it's actually going to fit together with the MHC receptor that's presenting that piece of antigen on its surface. So they will come together and they will fit together perfectly. Once that helper T cell fits perfectly with that MHC2, the helper T cell gets activated. Once the helper T cell gets activated, okay, so again, if the helper T cell goes ahead and combines with the MHC2, the helper T cell gets activated. And once it's activated, it will actually start releasing what's known as cytokines. And these cytokines are actually going to, those are going to help actually activate so many other immune cells that we have in our body. So one more time, helper T cells cannot directly bind to a pathogen. So they cannot directly bind to a pathogen found within your tissues, found within your plasma. They just can't. The only way they can go ahead and be activated is when they bind to the MHC2 found on one of the antigen presenting cell. It doesn't always have to be a macrophage. Remember we said that it could be a macrophage, it could be a B cell, or it could be a dendritic cell. Once they go ahead and they bind to that MHC2 that's presenting the antigen to them, that's when they get activated. And once they're activated, those helper T cells will start releasing cytokines. So this is an image right here kind of showing you the helper T cell binding to right here. Here's the helper T cell. And again, it has its receptors, and then it has also those CD4 receptors, and it will bind to one of the cells that have an MHC2 on its surface. And that MHC2 is presenting a piece of a foreign antigen, right? Because as we said, a macrophage or a dendritic cell or a B cell, they have the ability to engulf the bacteria or engulf that pathogen, break it down into little pieces, take a part of that antigen and present it on their surface. Once they present it on their surface, the helper T cell can go ahead and make direct contact. Once the helper T cell is activated, it will start releasing cytokines. These cytokines, they can actually help activate so many other cells. They can actually help activate B cells. They can actually help activate the macrophages themselves, the dendritic cells. They can actually help activate another set of T cells that we are about to talk about, which is known as the cytotoxic T cell. So actually our helper T cells are the most important cells within our whole entire immune system, because as you can see, the cytokines that they produce once those helper T cells are activated can help activate all of your other immune cells. And so therefore, if those helper T cells are deactivated, like they're not able to release those cytokines, your whole immune system is really going to be compromised. Like your whole immune system is really going to start shutting down. One of the viruses that's pretty harmful is the HIV virus. And the reason why the HIV virus is one of the most dangerous viruses out there, because the HIV virus attacks your helper T cells. And therefore, your helper T cells are not going to be activating your other immune cells. And so that's going to cause your immune system to be extremely compromised and extremely weak. The second set of MHC class that we have is known as MHC class 1, and those are receptors that are found on the surface of any nucleated cell. So any cell in your body that has a nucleus should have an MHC1 receptor on its surface. So let's say, for example, that this nucleated cell is a normal skin cell. That normal skin cell is going to have that MHC1 receptor on its surface, and what it's going to be presenting is a piece of its own self-antigen. So when we say self-antigen, that means that this is a part of the cell's antigen. It is not something foreign, so therefore we refer to it as self. And it's going to be presenting it right here on the surface within the MHC1. Now, this piece of antigen presented by this normal skin cell right here, it's being presented to another T cell known as your cytotoxic T cell, or we abbreviate it as CTL. Cytotoxic T cell also has another name. We can refer to it as your CD8 T cell. CD8 has a receptor on its surface, so it has a T cell receptor, and then it has another protein receptor known as the CD8. Now, in this case right here, the cytotoxic T cell is not going to attack that normal skin cell. Why? Because remember how we said your T cells are made in your bone marrow and then they go to the thymus gland so that they can become mature, and they really have to recognize the difference between a self-antigen and a foreign antigen. So once they become mature and they can differentiate between what's a self-antigen and a foreign antigen, this cytotoxic T cell right here is going to be able to recognize that this is your self-antigen, and therefore it is not going to attack that skin cell right here. So right here, no attack is going to take place because it's going to recognize again that this is your own self-antigen. Now, if we take a look at the second scenario, so let's say that again you have a cytotoxic T cell represented right here on its surface. You have the T cell receptor. Also on its surface, you have the CD8 protein receptor right there. And let's say that this is, again, your skin cell, but now there is something different about that skin cell. Let's say that it's either cancerous or let's say that it's virally infected. If you remember, we also talked about how viruses, in another video we talked about it, how viruses attack your cells and how they inject their own nucleic acid within your cells. So let's say that this is either a cancerous cell, or you can think of it as a virally infected skin cell. Now, what's going to be different about this cell is that the antigen that it's going to be presented on its surface is actually going to look different. I tried to kind of like shade it just to kind of, or outline it with a different color, just to kind of tell you that now this antigen has been altered. And what's going to happen is the cytotoxic T cell, when it's going to go ahead and take a look at that altered self-antigen, it is going to be able to tell that, you know what, this self-antigen doesn't look the same, and so therefore there's going to be contact between that cytotoxic T cell and that cancerous or virally infected skin cell. And the T cell receptor is going to bind with the MHC receptor, so there is going to be contact between the two, and the cytotoxic T cell is going to go ahead and attack. Actually, it's going to kill this infected cell right there. Versus in the top scenario, we said that as long as your cell is presenting a piece of its own self-antigen, then your cytotoxic T cell is not going to attack it and it's going to leave it alone. But if the cytotoxic T cell recognizes that the self-antigen has been altered, then it's going to make contact with it. It's going to bind with that MHC receptor, and it's going to release chemicals that will kill that cell. Those chemicals are going to induce the cell to commit apoptosis, which is basically cell death. So cytotoxic T cells, they're going to be able to kill a cell that's presenting an altered self-antigen on its MHC class 1 receptor. And as I said, it's going to inflict harm on the cell by causing the cell to commit apoptosis, which is cell death. Now, how does it do that? Once your cytotoxic T cell goes ahead and binds to the cell that's now been either virally infected or there's something off about this cell, it's cancerous cell. Once it binds to it, it's going to start releasing chemicals known as perforin. Perforin coming from the word perforate, which means that it's going to create holes on the surface of the cell. Then it's going to puff another chemical known as granzymes, and those granzymes are actually going to go deep into the cell and they're going to induce cell apoptosis. It's basically going to force the cell to commit cell death. So the cell is eventually, as you can see, it's going to start dying, kind of, shriveling up like this, and it's going to die. So if you compare helper T cells with cytotoxic T cells, helper T cells do not directly attack. Helper T cells, they just activate your other immune cells that can go ahead and attack. On the other hand, cytotoxic T cells, they are able to directly attack that cell that has been altered. Now there's another group of cells known as natural killer cells, and those natural killer cells, they actually act in a very similar way to the cytotoxic T cells. They're able to bind and recognize self-altered antigens on the MHC1 receptors, and they go ahead and they release chemicals that would induce apoptosis. So one more time, they induce the cell to go ahead and commit cell death. So as I said, the cell could either be cancerous, or it could be virally infected, or it could be attacked by parasite, again changing its antigen, and therefore again the natural killer cell is going to go ahead and be able to recognize that, and it's going to be able to destroy it. When we talk about acquiring immunity against a certain pathogen, we talk about naturally acquiring that immunity or artificially acquiring it. And under either group, we have active and passive. Active would mean that you are making your own antibodies and your own memory cells. Passive would mean you are given the antibodies right away. How can you naturally acquire an immunity against a certain pathogen and create your own antibodies? If you get sick. So if you go ahead and get sick, with like you've contracted a certain infection, then your body is going to actively start making its own antibodies. It's going to start actively making its own memory cells. How can you acquire antibodies in a natural way? If, for example, a mom passes on her own antibodies to her fetus via the placenta. In this case, the fetus is not making its own antibodies, it's actually getting it from the mom, and so therefore we refer to it as passive. Artificially, how can you actively make your own antibodies, and that would be considered an artificial immunity, is if you don't get sick on your own, but you get vaccinated. So if you get vaccinated, that vaccine carrying the dead virus or carrying the weakened virus, if it's injected into your body, you are going to start making your own antibodies against it. You're going to start making your own memory cells against it. And so again, you are actively making your own antibodies. Passive is, as I said, when you are given the antibodies. Let's say someone gets got bit by a snake. If they got bit by a snake, you don't really have that much time to make your own antibodies. Because if this is your first time getting bit by a snake, remember how I said it would take up to 10 to 17 days for you to make a good amount of antibodies. Well, you don't have 17 days if you get bit by a snake. You might just have a few hours. So this individual, like you would actually rush them to the hospital so that they can get an antivenom, so that they can get injected with these antibodies that will help them right away. Those antibodies, though, that are injected into your body, they're not going to last for a very long time. Eventually, they're going to degrade. Versus when you make your own antibodies, they're going to last longer. Again, they will eventually degrade, but they will definitely last longer if you make your own antibodies versus if you are given those antibodies.